My name is Jonas. And today we're going to tell you how we created a mycelium-based solution to basically get rid of bad odors in our environment and how we created an innovative odor-absorbing waste container with it.
So, in our daily environment, we often encounter undesirable odors. These smells affect our psychological and physiological health. There are not many ways to eliminate odors, and most of them are harmful to the environment. Often, people choose to mask negative smells by spreading other scents, which frequently doesn't improve the situation. Different smells mix, but the bad odor remains.
So our main goal was to basically create an innovative waste container that would be based on organic materials. And a few of our objectives were to basically select a natural material that could be used for odor absorption, to create a structural material for the waste container, to print it and create it, and also to test the effectiveness of our mycelium product.
So,
Okay, so let me tell you why does waste containers basically smell that bad? That odor is when microorganisms basically decompose organic waste that are in there. And that odor is primarily caused by protein breakdown and the decomposition of fats and food residues. And probably the most sticky compounds are ammonia, amine, sulfur compounds, and there are compounds such as CO2, for example, and they can, in high concentrations, they can be quite dangerous to our health. However, they might be quite inconveniencing even for our psychological emotions.
So, what is mycelium? Mycelium is a vegetative pod of nucleus consisting of a dense network of villi. So, it's the pod that is not seen with our eyes. The largest mycelium networks can reach up to 10 quadratic meters. So, what is important of mycelium that it has features such as large surface area and porous structure made of biopolymers.
So mycelium is probably one of the most popular materials these days in biotechnology. It is quite light because of its versatility. It is used, for example, by startups to create some insulating barriers, to create green packaging, to replace plastic, or fungi is sometimes used as a substitute of meat. And these days we still do not know a lot about mycelium, therefore a lot of studies are being done on it. However, it is considered to be a great alternative to plastic.
So, because mycelium is dense and porous, it can lock other molecules onto its surface. Even dried mycelium retains these properties, making it an excellent choice for suppressing bad odors. So, it really well absorbs nitrogen and sulfur compounds. Therefore, mycelium is perfectly sited for stopping waste and sweat odors in enclosed spaces.
So now we would like to tell you about how we cultivated the mycelium ourselves. So the process of preparation isn't that difficult. However, it does require a quite sterile environment. The most important thing is probably the preparation of a substrate. We use wood as a substrate to grow mycelium. And basically, it's very important to heat that substrate to remove all, for example, spores of mold in it or remove all bacteria because, mycelium is quite susceptible to any diseases.
So mycelium is grown in dark and room temperature, and when you grow it, you need to be aware that it is in clean surface.
Right, so here you can see some pictures through a microscope of, here the mycelium is already dried out. After growing it, we need to dry it out to stop its growth and to stop the spread of spores. In the second picture, we can see the hyphae here. It still has not been dried out. However, if we look very closely, it's made of these tiny like round balls onto which the molecules of bad odors can basically join and they can be collected. So to basically test the effectiveness of our mycelium, here you can see some pouches that we made. We'll talk about them further. And basically to test it out, we wanted to, of course, We tested it out ourselves and we noticed the results. However, we wanted to do a more scientific study. Therefore, we created this 3D container model. We implemented an MU135 sensor into it together with a Arduino All3 microcontroller. And it can detect, for example, levels of CO2, ammonia, and what's most important for us is the alcohol vapor gases because, well, they are released when basically organic material decomposes. And the study is currently undergoing. However, the first results are definitely okay. The first results are definitely good. Here you can see some pictures from the printing process of the trash can.
So, after investigating that mycelium can indeed trap others, we realized that this could be beneficial to both individuals and institutions, which led us to establish the company Branch Room.
And we wanted to make mycelium as accessible to everybody and as easy to use. Therefore, after some trial and error, we came to the idea of these mycelium pouches. They are very easy to use. You can just put them into, for example, you can hang them near a trash can, in the toilet. You can put them into, for example, a drawer or a sports bag, and they will remove post or odor from these environments. They are easy to use, they are replaceable, and basically after use, so they last about like a month and 2 months. After use, they can be completely decomposed, just thrown into, for example, a waste container, an organic waste container. It can be just thrown in general trash, and they will completely decompose because they are organic. So from that, we basically found out about all those are caused by the decomposition of organic materials. We successfully grown and applied mycelium, and using the 3D printer, we created a waste container with a dedicated space for bag and sensors. And to summarize everything, we successfully developed an organic mycelium J solution that basically absorbs all those from the environment, and we successfully created our own company.
So, where you can find us, we have our Instagram and Facebook. And also you can find us on internet on branchroom.ld.
Right, and we wanted to end with quite an inspiring quote by Leonardo da Vinci that nature is the source of all true knowledge. Thank you.
Station may we have some questions?
Yeah.
Do you have some samples?
Yes.
Here.
Yeah.
That's interesting. How long does this are using? How long is the production process?
Yeah, of course. Here we can have out some samples. Here, right? So the process of making them isn't too difficult. Mycelium is grown about like 3 weeks. And well, the most difficult is probably prepare. They are made from linen, we hand saw. And
Yes, I saw.
yes, yeah, exactly. And well, it isn't too difficult to prepare them, but from the start till the end, it's probably about 3.5 weeks.
Have you established a strategy?
Yes, exactly.
And how did you go to the store?
Yes. When, for example, in the Christmas Expo, we were nominated as the best on-startup business idea in Lithuania. Last week, we were nominated as the best company in the Klaipėda district and the surrounding first districts, and we also were nominated as the best option for education.
Thank you.
How much does one bag cost?
€6.20.
But of course, we offer, for example, subscriptions or buy them in bulk, and then the price is lower, of course. Okay, thank you.
One question.
Okay.
Thank you. That was a really good presentation. Well, it inspired us, right? Yeah, so thank you really much. So, shall we go to the next presenter? Are you ready? Okay, then I want to invite our friends from Latvia. So, Airita Ešvalde, yes? I'm a teacher. You're a teacher. So, the girl name? Can you present yourself? Please come. Oh, sir.
Hi, my name is Liga.
My name's Eva.
We are from Klimatui Skola, so the Lithuania and Latvia. So, last year, our school participated in a global project. The goal was to raise awareness about water pollution. It causes an effect on the ecosystem and human health, and to encourage students to take action on the pollution and protect our water resources. Of course, a big thank you goes to our teachers for writing a project called Water Quality Monitoring the Water River. There is a small river called Mėlyna River that flows past our school, connecting with the Vatlaya River, which is a tributary to Box River, which then flows into Liepājas Lake and then it connects to the Baltic Sea through Rotterdam. Last spring, on 24th of April and 27th of May, 5th and 7th graders went on a research trip around Liepājas Lake with a floating water jury activity. Students studied water from oxygen and chlorine levels, as well conducted water quality research and observed nature. They collected water samples measured different indicators as pH and transparency, and learned how pollution can affect aquatic ecosystems. During the trip, students also identify various plant and animal species living in and around the lakes, gaining a better understanding of the biodiversity. The activity helps students develop practical skills and increase their awareness of environmental issues, especially water pollution and the importance of protecting natural water resources. Also, on May 27th, 8th grade students assessed water quality of Mėlyna River at 3 different places. They learned how to use determiners to recognize plant and animal species, as well as how to measure the river's flow speed and depth. Based on their observation and collected data, students analyzed environmental indicators and drew conclusions about the overall quality of the river. They also prepared detailed research protocols using both their reasoning and biological indicators such as aquatic plant and organisms to score their findings. On June 26th and 27th, 7th and 8th grade students went on a rowing trip along the Mėlyna River, where they learned about the plants and animals found in and around the river, as well as explored the river environmental firstly. The following day, the students were introduced to a profession of hydraulics with the hydrological station. There they learned how water levels flow, how quality is monitored. This data is used to assess environmental conditions and manage water resource responsibility. On the 2nd of September, our school didn't just study in school. Our school organized a water day. This day was dedicated to teach and learn about water and its importance. So kids from first to third grade learn about water safety, explore the concept of vitamin U, and enjoy laboratory water show. Students from grades 4 to 6 participated in water painting activities, learned how to filter water using natural materials, and attend a lecture about the water cleanliness and pollution. Meanwhile, from grades 7 to 9 and 12, part of educational activities led by the National Museum of Nature. They also attended the lecture water show using the message making station, and they created infographics on the topic how to save water, as well as about safety near water and encouraging awareness for responsible water use, environmental protection, and safe behavior of rivers, lakes, and the sea. On the 17th of September, students from 8th and 9th grade traveled to Klaipėda, where they visited the Lithuanian Sea Museum and the Klaipėda Dolphinarium. There they learned about marine plants and animals, their habitats, and the importance of protecting sea ecosystems, including the impact of pollution in life. Under students created information board next to the river where gathered presented information about the river, as well as the plants and animals living in and around it. They researched variable species, selected the most relevant facts, and organized concept in a clear and engaging way. Students also translated the text in English so it's more accessible to tourists. This activity helped develop their research and language skills while promoting awareness of the local river environment and the importance of protecting natural water resources. Throughout the project, we learned about river pollution, connect back to the parts and living the environment. Assessing the water quality of the river, also collected and removed trash surrounding area. We made sure to work responsibly and we were careful not to leave any waste. Altogether, we as students who participated learned a lot about water pollution, and we continue to learn and teach others about it. Through different activities, research trips, and hands-on work, we gain deeper understanding of how pollution affects rivers, lakes, and seas, and why it is important to protect water resources. As well as we checked out our local river.
Well, may we have questions from audience? No, because what did you what did you learn about water pollution project this
A lot of water pollution happens from us humans mainly. When we went on the rowing trip as well as made infographics and protocols about our river close on school, there was a lot of trash. It was mainly about the students and the people who maybe go fishing or there's a bridge that goes across our river that throw trash into it, which made us sad. We also collected it.
why do you think people don't trash?
Because humans are lazy and they're I don't know, they're lazy to go to the trash can and just
Let me ask a question. What was the most shocking fact that you discovered during your analytical?
into our river there is sewer that goes from our homes like from toilets and that. We didn't know that was also breaking. We told about it, but we couldn't remember.
Yeah, sometimes we talk a lot about the problems, but we do not know where they come from. And I think that was a really interesting adventure for you, right? Because you traveled all across the river, you just seek for more information, so you learn a lot, not only about the water, but again about ourselves and all of your environment. So thank you. Yes, and for your presentation, we have a few gifts. Yes, one. Thank you. Thank you so much. Really good. Okay. So we're almost halfway through. How are you feeling now? Great. Cool. Why so sad then? Because City has already presented your ideas. So I think, yeah, we're almost near the end, but again, your presentations are really, really interesting, and we are just waiting for more. So I think we should start the third presentation, right? Okay, so let's invite to present the idea of Urte Urbonaitė and Agota Juknevičiūtė. You'll do okay. Smile. Don't worry, smile.
Hello, we are Urte and Agata. Today we will present our project, Effect on Filtration, Water pH Stability under Increased CO2 Exposure. Our research aim was to investigate how different water types respond to acidification and evaluate their buffering capacity. Our hypothesis was filtered water, especially with carbonate layer, will show a smaller decrease in pH when CO2 increases. Water acidification happens when CO2 dissolves in water and forms carbonic acid. This lowers pH and can be harmful for aquatic life. That is why it is important to understand how stable water pH is. We tested two types of water, seawater and perennial lagoon water. We had two conditions, normal air and increased CO2.
We measured pH before and after 24 hours. We also measured CO2 concentration above the water using a sensor.
We used two filters. Filter 1 had cotton wool, carbon, sand, and gravel. And filter 2 had carbon, sand, gravel, and clay. We want to see if filtration changes pH stability.
After 24 hours, pH increased in most samples. We saw similar pH changes in both CO2 and normal conditions. Filtration did not have a very strong effect on pH.
From the graphs, we can see that pH values stayed similar or increased. There was no clear decrease because of CO2. Filtered water was slightly more stable, and overall the differences were small.
CO2 levels were similar in all samples. This means all samples had similar exposure conditions, so our results are reliable.
The data shows that pH changes were small in both water types. Sometimes pH increased instead of decreased. Filtered water was a bit more stable. Seawater showed a little more variation than liquid water.
Our hypothesis was only partly correct. CO2 did not always decrease pH. In some cases, pH stayed the same or increased. More research is needed to understand why.
To reduce this problem, we should reduce CO2 emissions. We can use natural filters like sand and carbon. We should protect water ecosystems, and small actions can help the environment.
At the end, we have a short video to present our experiments and results.
What if the water we depend on is slowly becoming unsafe? As carbon dioxide levels rise, more of it dissolves in water, forming acid and lowering the pH. If no action is taken, this can harm aquatic life, damage ecosystems, and reduce water quality. In the future, it may also affect human health. So we looked for a simple solution. We explored whether natural filtration could help reduce these changes. Some materials, especially mineral-rich ones, can neutralize acidity and stabilize pH. To test this, we created small laboratory-scale filters. We used cotton, activated carbon, sand, gravel, and carbonate materials like crushed limestone. Then we tested sea and lagoon water. Each sample was exposed to increased CO conditions, and we measured pH changes before and after filtration. Our results showed that after filtration, the pH sometimes increased, meaning the water became less acidic. However, the results were not consistent, and a clear effect of the filter could not be confirmed. Still, filtered samples often showed more stable pH values, which may suggest a potential stabilizing effect. This suggests that simple filters could become a useful tool for improving water quality. But more detailed research is needed to better understand their effectiveness. The problem is real, and this is only the beginning. So the question is, will we do more to protect our water?
Thank you for your attention.
Thank you for your presentation. May we have questions from the audience?
So, what's the point of using a water filter?
To clear our water, to make it less acidic and to balance our pH levels. But you also mentioned about the reduction of the CO2. May you know what could be the examples, how to do that? Because not all of us can make just filters to drink that water. Just by your environment, what you do. Know how to help you. Okay, maybe by just going to school or somewhere. Oh, yeah, of course we have the beautiful beaches there, right? But what to do that would, you mentioned in a video that we have a lot of animals that live in the Baltic Sea. But again, what just to do so we could save even for a bit about this happen. Because you just said really beautiful things.
You can make bigger filters to clean the water. And I don't think like a person by going to school can do anything. We have to make some big changes for this because this is a big problem, and the problem has increased over the years. So I think to help it, just a person going to school, he can help it. We have to like do it like together. Together, yes. It's
Yeah.
a bigger problem, so every person can do it.
Yeah, but actually, even when you're alone and you're thinking about the solutions, you actually can change the future just by talking to people, just saying what to do, maybe to motivate some other peoples that you know surrounding all of you. Because when we are thinking about the Baltic Sea, our rivers, oceans, lakes, it's not only just because of the water, because there are even more things that can come to that water. For example, yes, we have a lot of cars. And again, we have a lot of sewages there under the roads. Again, everything can go down to the drain and reach the Baltic Sea. So you show us a really, really beautiful way to dilute the water, but actually there are even more solutions. And I think that some of you are actually even doing at the moment in your schools, maybe in your homes, some of the best things that can change the future of our Baltic Sea and even the whole environment. So thank you really much. Just don't escape. I have a few things for you to give you more motivation. So thank you. So yeah, now we are halfway through, but again, I just want to say thanks for all of the presentations because all of your ideas are really, really beautiful. But again, just to make these things work actually needs a lot of our help, a lot of our motivation to do that. But actually, when you are thinking about the future, because here in the museum we say that 2050 can be really crucial because that year can change drastically from our behavior at the moment. Because we think that we are living in that really free world and we can do whatever we want. But actually, we have to think about the future and what to do. What are the solutions? But again, you showed us that you have really, really great solutions. So I hope that all of the animals and all of the people later, maybe even today or even after many, many years, will say a huge thank you for your innovations and these beautiful presentations. Okay. So, shall we go to the next presenter? Okay. Guys are nervous now. Will you present? Oh, that's right. Okay, so let's come here, and now we will have a presentation from Martas Baltieros and Dominicas Munoz.
Yeah, my name is Dominicas, and this is today. Today we're modeling the potential of making the water safer in their quality, and trying to save other species of fishes.
Yeah.
Context. Changes in fish populations in the Coronian connect to changes in water. Or how the changes in CO2 affect water pH and the possible conditions for microorganisms. To investigate how increased CO2 concentration affects the pH of the Coronian lagoon water. Increased CO2 concentration lowers water pH, which might negatively affect aquatic organisms.
So in regards to what is it? So in over these years, there's been a decline in fish populations, and there's less and less we can find in the lagoon. So our solution is to research about the water's quality and the pH levels to see if that's the reason why fishes are leaving the lagoon.
Russian fish population plan in the Crimea lagoon may prevent changes in water quality, CO2 or water pH, which can affect aquatic organisms. This study models how CO2-driven pH changes might influence aquatic life.
So we decided to take six for four samples from four different places and put them in 6 cups. So 3 cups got with more CO2, so we did three equal breaths in it and really closed it. And the other ones didn't get anything, just casually closed it. We put it in the same room, same temperature for 7 days and studied their pH levels and CO2.
The graph shows how pH changes in water samples after 7 days with and without CO2 exposure. In most samples, pH changed only slightly, and the results were not consistent across all samples. This means that the effect of CO2 on pH is not clearly observed in every case.
So the CO2 levels after 7 days were also inconsistent. Sometimes it increases, sometimes it decreases, sometimes they just don't move at all. It's just very inconsistent and there's no clear pattern for it.
The results show that pH changes were generally small and well controlled in CO2 treated samples. In some cases, CO2 samples showed a decrease in pH, but this was not consistent across all samples. The effect of CO2 on water pH was not clearly observed under the conditions of this experiment.
So we made a map of places. So the red circle is the places where we took the samples from, and then we examined it, and now we see this one is pH levels. Did it decrease? Did it stay equal, or did it increase? And CO2 after 7 days, did it still less or more than 2,000?
The water was only partially supported as pH changes were small and inconsistent across all samples. CO2 exposure led to slight changes in pH in some samples, but no clear trend observed. Changes in pH affect aquatic organisms, with further investigation may be expected.
Since we're implementing like an electronic fishing log that will keep count of every single fish species, how much and when and what place they were caught, into one app, which will also help monitor fish population, prevent illegal fishing without any license or permission, and detect changes earlier before it's too late.
Restore project review. The study demonstrates how CO2 influences water pH levels important for.
Hello, today we're discussing about the problem in the Goo Lagoon about fishes population going more and more extinct every year. The situation, we took the sample from the Danas Upe, but still it probably will be the same. So our situation here of these fishes are that there's a mass fishing, water is getting more and more polluted over the years, and also that there's less and less time for fishes to repopulate. So our solution to fix these problems are simple. First, destroy the barriers of fishes getting into here to migrate. Basically, they're having a really bad time getting through there from NEC to here. So maybe we should destroy the barrier. Second, we can save their homes from being destroyed. Make sure to keep the water clean, and fourth, limit fishing to a specific season and how much fish you can gather in a year. So I hope this situation explains, and I hope you listen to the problems. Thanks.
Thank you for your presentation. May I ask a question? Because you mentioned an app, right? But how do you imagine how that app would work?
Well, so so the person logs in into the account and has to keep count of the fishing. like they have to write have to find the right species, the amount, and when did you find. But then it will be thrown into the log to the log of the internet and it'll be monitored.
So it could be useful to maybe talk with our other fishermen or just simple people just love to maybe fish some fishes. But don't you think that maybe it could lead to fishes amounts decoration of the fishes mouth more because people maybe won't use that. What are your suggestions?
Good.
But I think that's a really, really useful way to count how many fishes are still left in our Coronian lagoon. But I think that we have even more things to talk about. But an app is really, really good invention. I think that even our younger generation would use that app even more, right? Would you use that? Yes, because we all will do. So thank you for your presentation. May we have questions from audience? Oh, so you will use the app, right? Okay, so thank you. So I just want to mention a few things, maybe a little bit more about the Baltic Sea, because actually we all know that, yes, we had a lot of world wars in our Baltic Sea region, but actually after Second World War, we have even more shipwrecks that are still in our bottom of the ocean of our Baltic Sea. We have a lot of other types of pollution that is still happening in the Baltic Sea, but actually that's pollution of the water, even the the quality of the water is actually changing, but sadly the Baltic Sea, as our professor from Pakistan just mentioned, sadly, our Baltic Sea can clean itself, but we pollute it more than the sea can help itself. So actually, it depends on us how that future will be. So I really am really grateful for your inventions.
So if you go to the second slide, you would see that we had different goals for different age groups. So for the 5th year, we had the water quality, and for the 6th year was a DNA. For the 5th year, we also had a workshop for the primary school students. And the start of our water quality for the 5th year starts at slide 3. We measured the physical chemical properties of water, and we used simple test kits and recording templates so that we could test and record the properties of the water. And we sampled these in March and May. Those were the main monitoring times. And we also had an extra one in September, which we did this year. We also did some biological sampling in May and September. As you can see on the picture, the yellow zone, there is where we sampled the biological sampling. And we also noticed that the wind has a big effect on the organic load, and it pushes it to the blue and red sides of the pond, if you could see that on the picture as well. And if we go to slide 4, we would see the results of the water properties. We see that ammonium and nitrate have peaks in May because of the geese and ducks that are surrounded there. But in September, we see that the ammonium drops, and all the nitrates and nitrate in the water is below 1 mg liter in May, and is slightly more nitrated, is being converted into nitrates. Yes. So at the other side, we also see another chart. This one talks about, or states, no, the other slide, please. Yes, we also see that there's phosphate. We see that the phosphate decreases, but at first it starts taking the lead, but in May and September, we see that the values are dropping slightly. And in the next slide, we did measure the O2 and CO2, and we saw that oxygen is very important in the ponds, but because of construction work, the oxygen levels went down, so the organisms in the water weren't really as much as we thought there would be. At the next slide, we see the biological sampling, also known as the BBI. We used a chart. I thought I was going to bring the chart over, but I forgot. And we had a chart to show how we calculated the BBI, and we ended up having BBI 7, which indicates that our water quality, we have a good water quality, and our pond is doing quite well. For the next one, we have a triangle, and this one shows us the food web. So at the bottom, we see nutrients such as phosphate and ammonium that are eaten by algae, larvae, and different kinds of organisms. These are eaten by other larvae and microinvertebrates, which also gets eaten by the duck mussels and frogs and stickleback fish. And the stickleback fish gets eaten by the heron and other species from the pond. But the algae also gets eaten by the Canadian geese and ducks. And if these organisms perish, they all become nutrients, and the food webs will start over again. So it's a cycle, kind of. At the next slide, we'll see the environmental pressures. Because of the construction works, we had heavy sediment load in September, which made it quite hard to do the sampling, but we tried to do our best and it went well. We also, because of that, the water level wasn't really stable, and the water temperature was also rising. So that was quite pressure for us to do the sampling. We also had some rock salt. There was also rock salt because of the construction work they were doing nearby in the playground we had in the winter. Next, we'll see that we have DNA techniques. Our samples were taken in March and September, and we used bricks to collect organisms. And we also did tissue construction, such as muscle tissue from the duck mussel and frogs around our pond. And we also had 3 main steps we did. The first one was DNA extraction, where we used a lysis buffer, Putanes K, for the animal species, and because of these, we could do our project with no problem. The second step was PCR application, also known as DNA application, where we used polymerase chains to see the reaction. And our last step was the gel electrophoresis, where we isolated and we saw different bases and pairs of DNAs, where we also could see that we also could make something out of it for our project. So we also saw that different species also equals to different protocols. So we used different protocols for every step we took. And if you go to yes, this one is the DNA techniques we just talked about, the lysis buffer and the DNA extraction, how we did it. We used the lysis buffer and the Putanes K. So firstly, we broke off the cells and to release the DNA so that we could use the DNA and not the cells that were left. Next, we used a centrifuge and we purificated the DNA so that we could go to our last step, which is heat treatment, and we used the temperature was about 60 degrees Celsius. And because of this, we could avoid contamination as well with the proteins that we treated by using a spectrophotometer at 230 nanometers and at 260 nanometers, which all went very well. If we go to our last slide, we will see that there are different calculations. We calculated how much CO2 our odors actually makes. So if our odor comes from abroad, how much CO2 it makes from abroad coming to our country. We calculated that, and we saw the amount we calculated, we compensated that with trees. So we planted some trees at our school, like the willow, oak, the walnut, and some apple trees, to compensate the CO2 we made in the past period. And as for the 5th year, we really enjoyed this project, and we hope we could do more in the future. Thank you.
Okay, thank you very much, Rosalind. And I'm going to just show you the audience real quick so you know you weren't speaking to yourself.
Hello.
So, thanks so much for joining all the way from Antwerp. It's a very beautiful city. I love it. And we hope that maybe we get to see you and visit you weekend Erasmus.
I hope so too.
Yes, and keep your good work.
Amazing research.
Thank you.
Bye.
You too. Bye-bye.
So, okay, so we had some technical issues, but still we had a really, really wonderful presentation. But at last, we are at the last presentation. So are you still up? Yes, okay, so I will invite to present Gintare Batskulite Ilya Ordenai.
Yes.
Programoje veikite, tai tiesiog pasakykite, kiek suprantama ir įmanomas.
Good day, everyone. Today we will present our project called Assessments of Eutrophication Risk in Water Bodies based on chemical parameters. We are students from Klaipėdos Varpas Gymnasium, and this research was prepared as part of the project Sea Change, Students for Carbon Neutral Coast.
The aim of our research was to analyze chemical parameters such as nitrate, phosphate, and ammonium in different water bodies. We also aim to indicate the potential risk of eutrophication and consider pH changes under CO2 discovery. Our hypothesis was that water bodies with higher nutrient concentrations may indicate a higher potential risk of eutrophication. We also expected that the samples would show greater pH changes after CO2 exposure.
Eutrophication is the excessive growth of algae in water due to high nutrient levels, especially nitrogen and phosphorus. It can lead to oxygen depletion, harming aquatic life, and reducing water quality.
We collected water samples from the Baltic Sea, Curonian Lagoon, and Vanya River. We measured nitrate, phosphate, and ammonium concentrations using test kits. pH data were obtained from the
Samples were collected from several locations. Sea samples were taken in Karthe and Melnaya, lagoon samples from 4 different points, and river samples from the Vanya River.
The result showed higher nitrogen concentrations in lagoon 4 and river 1. Phosphate levels were similar across all locations, while ammonium concentrations remained low. These results may indicate a higher potential eutrophication risk in some lagoon and river samples compared to this.
pH changes were small in all samples. No clear effects of carbon dioxide exposure was observed.
Based on our results, higher potential eutrophication risk was identified in level 4 and level 1. These locations are closer to human activity, which may explain higher nutrient levels. Moderate risk was observed in some lagoon and river samples, possibly due to their location in the shore or in more close areas. Low risk was found in most sea samples where water mixing and dilution was strong. Higher nitrate concentrations in some samples may be related to local human activity, such as runoff from nearby settlements or urban areas. Phosphate levels were similar across all sites, which may indicate a more uniform distribution or limitations of the measurement method. Ammonium concentrations were low, possibly due to rapid conversion into other nitrogen forms. pH changes were small in all samples and no clear relationship between carbon dioxide exposure and pH changes was observed. Our hypothesis was partially supported. Higher nutrient concentrations were associated with a higher potential eutrophication risk. However, greater pH changes in the CO2 exposure were not observed. pH changes were small and the relationship between CO2 exposure and pH was found. Further investigation is needed to better understand the results. We propose regular monitoring of water quality and reducing pollution sources such as sewage discharge, and household chemicals entering water bodies. Reducing nutrient inputs, especially nitrate, may help lower the risk of eutrophication. Improving wastewater treatment and controlling runoff from urban areas are also important. We also created a short video to raise awareness about purification and water pollution. The video explains the problem, its causes, and its impact on water ecosystems. It is designed to be shared on social media. For your attention. We hope our research helps to better understand eutrophication risks and the importance of protecting water ecosystems.
We have questions. Okay, no questions, but I still wanted to say that eutrophication, yes, it's a really, really important thing for our sea. As a lecturer from Pakistan mentioned, about 97% of the Baltic Sea is affected by eutrophication. So I hope that your presentation made a huge, huge impact on our awareness that we should do something that can be really, really simple steps forward just for the brighter future. So thank you, and of course, a little.